Thank you so much, Lena, and thank you all for listening to me on the third lecture. And as Lena said, coherence in the X-ray region is quite new. And when we combine it with coherent imaging techniques, it's allowing us essentially to make the first perfect X-ray microscopes. Every synchrotron, every X-ray free electron laser is actually using these techniques because it is the only way to do very high resolution imaging using X-rays. This is a 11-year-long project for us. It was started by a grad student, former grad student, Richard Sandberg, who's now working at Los Alamos, and then Matt, Bosheng, Dennis, Dan Adams, Liz, Chrissy, Michael, and Xiaoxi then, carried it along so we could, try to refine these techniques to make useful microscopes. Just sort of the interesting context to put this in is that when coherent imaging was developed for synchrotrons, you would get a week of time every 6 months. So you would go back to your lab and spend 6 months figuring out the image reconstruction. But we started doing it with the tabletop sources, and then we could take data much quicker. So even though we didn't have the same spatial resolution because our X-ray wavelengths were not as short, our data rate was a lot bigger. So we started having to deal with, well, how can we use GPUs? How can we make this feel like a real microscope? And that's still work in progress. So, but I'll tell you about it. So it's this topic here. How do we make functional imaging work? Because, for the most part, most coherent imaging has been of static structures, and even that was taking a long time to reconstruct the image. If you start thinking about if you want to put, a cell or a small, a test system, a nanosystem, or a functioning material, then you have all kinds of challenges that you're going to have to deal with. And this is not, this challenge is not confined to X-ray imaging. If you think about it, every area of imaging, from the beautiful super resolution imaging to electron imaging to X-ray imaging, they've all undergone really amazing advances in the past 5 to 10 years, every area. But the one interesting aspect of all these imaging techniques is none of them are anywhere near their physical limit. Some techniques are good for high time resolution, some for high spatial resolution, but none of them are the perfect imaging modality. And they're all way, way too slow. And a group of us got together because we wanted to try to propose a science and technology center. And what we realized is that we were all dealing with exactly the same problems, big data, algorithms, computation. And we were all going to different conferences, so we usually never talked to each other. And then we realized this might be a good area to propose, because it was a case where the same challenges were being addressed by completely different scientific communities, because usually, as Marcus Raschke, who's in my department, would say, usually it was about the electron imager saying, this is my image of the sample, and this is right because electron imaging is better. And then the X-ray folks would say, well, this is my image of the sample, and it's right because X-ray imaging is right. But of course, the actual truth is that, every imaging modality gives you a different view. When you compare them to each other, then you're actually, learning something about the sample, and you know what modality is good to see different things. Okay, so what we said is, okay, let's take the advantages in electron imaging, and it's just amazing because the same computational and software tomographic techniques that were developed for X-ray imaging, and the same advances in detectors that were developed initially for synchrotrons, then got applied to electron imaging. And that's why you have this amazing advance in electron imaging these days, where literally, this is work from Jiang Yao at UCLA, he has identified, nanoparticles, and this is one example where they could take a 7 nanometer nanoparticle and identify the positions of over 26,000 atoms and know exactly which atom was which, to be able to see defects, screw dislocations, just amazing. And this is work by Hang Zhu at UCLA that revealed genome transcription inside a virus with really high spatial resolution. So, there's a paper in Nature and Science almost every week in one of these electron imaging techniques. Nanoimaging is also amazing because it's a great surface imaging technique, and the interaction of light with the nanotip allows, makes it possible to image with chemical, mechanical, tomographic, and really identify the couplings in all kinds of materials. X-ray imaging, just in the last 5 years or so, we finally have learned how to build a perfect microscope, and so all the microscopes at the synchrotrons and X-ray free electron lasers are being changed to throw out the lenses and be able to enhance the spatial resolution. And of course, the other thing that's changing imaging is computation. That faster algorithms, ability to transport data, better sources and detectors, all of these are common. And what we said in our center, that we need to combine them, develop the underpinning technology, and then image common sample systems and common samples, either in the same microscope or sequentially, if we really wanted to find out everything about a nanomaterial. And so one gets asked to make a single slide, and I felt like they were asking us to make a single slide of the last 2 years of your life, and so we tried to pack a lot of things in. But essentially it was, so what are the hard things to image? As Lena said, if your sample is not crystalline, then it's hard to image because you really need to take a full field image. You can't assume periodicity. And so non-crystalline structures, we'd like to be able to, for example, image energy flow across interfaces, fast 3D atomic resolution imaging of biological complexes, understanding, for example, the nature of glass, or functional imaging of, for example, the systems I was talking about yesterday that are very fast charge and spin dynamics, or designer photovoltaics, or designer nanosystems where you can control the properties and the mechanical and the magnetic and transport properties. And for all of them, these questions, you can't achieve that capability with one imaging technique. So we have to try to combine electron, X-ray, and advanced nano-optical imaging. But of course, we want to build microscopes, but if you don't have some way of validating your imaging technique, then it's not going to be so useful. So this is the idea, to develop the underpinning technologies and then apply them to different techniques. So our tagline is microscopes of the future. We're building the microscope that you will need in your lab, let's say 10 to 20 years from now, and also push different imaging modalities to their limits and then validate. Of course, this is not something we can do alone. I wanted to show this picture because Naomi is the lead at Berkeley, and Naomi got her PhD working with Lena here at Harvard. So this is Naomi Ginsberg. She's the lead on nano imaging. We have an X-ray imaging team and electron imaging team. John Miao is the lead at UCLA. And then Laura Waller at Berkeley Engineering, who's the lead on underpinning technologies. And of course, we also have many activities in education and knowledge transfer. And just to give you an example of the really stunning advances in electron imaging, this is work from John's group from last year. And what he's using is electron imaging to take an image of this nanoparticle. It's an iron platinum nanoparticle. And it's a material that's being developed for data storage. But there was a quandary about this material. It was supposed to have a really high magnetic anisotropy, but when they would make the nanoparticles and then test, the magnetic properties were never as strong as people expected. And this work actually solved why that was true. That when they made the electron image, and then I wish this was from our microscope, but this is a simulation from the data. Okay, but this is what we want to be able to do in 10 years. So, but now they can take the 3D tomographic reconstructions and then pull apart the nanoparticle. And what they found was there were many different phases present in the same nanoparticle. Only one of them had the high magnetic anisotropy. And so that was why the material, everybody expected it to be great, but it wasn't. It was because there was not tight enough control over the synthesis. Okay, so for the rest of the talk, I'd like to talk about imaging using X-rays. And as I said in my previous talks, X-rays are great because they can, in principle, penetrate visibly opaque objects that are thicker than can be probed with electrons. But a big problem with using the very short wavelength of X-rays until very recently was the fact that you could not make a decent X-ray lens. A zone plate lens for the X-ray region cost between $50,000 and $100,000, and it could disintegrate in your hand as you walked across the lab. So one can afford this at the beautiful synchrotron sources because you have a whole infrastructure to do the X-ray imaging. But in a regular university lab or characterization lab, it's hard. It can be done, but it's hard. But on top of that, the best spatial resolution one could achieve, this was done with 2 nanometer light, so between 6 and 10 times the diffraction limit. So, and in fact, this was done in one place in the world, and more regularly, if you ask, if you want to buy an X-ray microscope from Zeiss, what spatial resolution can you get? It's, an order of 80 nanometers at best. And a really small depth of focus. You have to have a very stable lab to operate these microscopes. Okay, so fortunately, just at the same time when coherent X-ray sources were becoming available at high brightness synchrotrons, at the X-ray free electron lasers, and with these tabletop sources, a scheme that was proposed by David Sayer way back in 1952 became possible. So what Sayer proposed, well, if you have a coherent beam of light, you shine it on an object, collect the scatter pattern, and in a normal microscope, you'd put a lens in the scatter pattern, and that would take the Fourier transform, recover and make a real image of the object. What Sayer proposed is, okay, so you know the amplitude of your Fourier transform, but you don't know the phase. Well, why not use a computer algorithm or a phase retrieval algorithm at the time to recover the phase? And you can do that if you put some kind of constraint on the experiment. And the initial constraint wasn't great, but it's a nice one to explain the idea. The initial constraint was, let's just image isolated objects. Now, that's a very restrictive microscope, but it's what they did until 2013. So if you have an isolated object, and you collect the scatter pattern with your detector, you have no idea what the phase is, but you can assign a random phase and make the Fourier transform. And of course, you won't get an image of the object because you've assigned a random phase, and so it's incorrect. But if you know your object was isolated, then if you recover object in a region you know there was no object, you know that's an incorrect guess of the phase. So you can set these amplitudes here to 0 and take another Fourier transform. Now you have a better guess of the phase. You can replace your retrieved amplitude with your experimental amplitude, and you hope if you repeat this multiple times you'll converge. Now, a lot of the times you didn't. This initial constraint isn't so strong, but the wonderful thing about it, though, is that there's no optics between your sample and your detector. So that means that you should be able to reach diffraction limited imaging. You get a phase and amplitude image contrast, very robust to vibrations because the CCD pixels might be 13 microns, and you just have to have your microscope stable to microns, but you can still image at nanometer scales. And it is the most photon efficient form of imaging, which is important for X-rays. X-ray damage often limits the spatial resolution you can get. If you don't waste any photons, you can push the resolution limit. And the nice story here is that, so Sayre proposed this technique in 52. It was demonstrated by Jiang Miao as part of his PhD thesis in 99. So it can take a while. And the other cute thing about it is that David Sayre was a co-author on this paper. So that was really nice. Okay, so that was the paper. You know, we live in a different time now. We don't get away with publishing something so short, and we would not get away with the last sentence in his paper, which is the extension to 3 dimensions is obvious. So, okay, so how does this work? Okay, so, and does the phase matter? It does. So let's take an object, a fairly low contrast object, so a bunny surrounded by some blue. We take a Fourier transform, so we know we're doing this on a computer, so we know the amplitude and phase. And now let's throw away the phase, keep the amplitude, and make an inverse transform. We'll just set the phase to be uniform everywhere and see, do we get a bunny back? And we do get something back, and you might think those are ears, but it doesn't really look like a bunny. Now let's do the other thing. Let's throw away the amplitude and keep the phase. Set amplitude equals 1 everywhere, but we've kept the phase. And now you can see we have 2 bunnies. There's a ambiguity on direction of the bunny, but you can solve that in other ways. So the phase is everything. And so if you have a good algorithm where you can guess the phase, you have and of course we have the amplitudes, we do keep the amplitudes. This can really work well. But when using that simple isolation constraint, because it was not a super strong constraint, what people used to do is that they would get the scatter pattern and they would run the phase retrieval algorithm about 200 times, and then they would average all the reconstructions and they'd present to say the material scientist, so this is an average image of your sample. But of course, no material scientist wanted an average image of the sample. And this, and it was only in transmission. But all of that changed about 99 with, conceptually, you wouldn't, intuitively, you wouldn't think it would make such a big deal. But if you scan over overlapping areas and then force the algorithm to come up with both amplitude and phase of the illumination and amplitude and phase of the object, if there's 50 to 80% overlap, it turns out that that's so over-constrained that now you can sort of implement what you would call a real microscope, where you're scanning over a surface, you don't need isolation, and it is really changing this coherent diffraction imaging. It's a field that's famous for its acronyms. So this one is called tychography. And now essentially people are very, very serious, that it's taking over every synchrotron, every free electron laser. Many companies are now implementing this tychographic imaging. So this is a little cartoon of how it works. You scan your laser beam with overlapping images and then feed into the phase retrieval algorithm, and then you recover an amplitude and phase of both the illumination and of the object. So it's really nice, and then you can reach diffraction limited. And in fact, So we started in 2007 with Richard Sandberg's thesis, and the spatial resolution that we were achieving was nothing to write home about. 200 nanometers. I mean, you could design a good lens that could get you to that spatial resolution if you had a good illumination beam. But now we've driven it down to sub-wavelength at 13 nanometers. We worked there because it's the wavelength used for the next generation of EUV lithography. So there's fantastic mirrors available there, detectors. There's a lot of technology. That wavelength was picked because you could make good wavelengths and detectors there. And not only in terms of the spatial resolution, but in the quality of the images. You can see this was John's first image that he made in 1999. It's the first two-dimensional coherent diffraction imaging. This was the first image Richard made for his PhD thesis. So you can see all of these are toy images of just holes punched in pieces of foil. Yeah?
I'm sorry, this is a stupid question.
No, there's no stupid question.
So I understand how you get the intensity with just the standard
Yes.
detection, but how
Yes.
exactly are you extracting phase?
You're using a phase retrieval algorithm.
All right, so that's not a direct measurement.
It's
You have to rely on computation.
yes, you do rely on computation, but the accuracy of the image does depend on the algorithm you use, and if you've over-constrained the algorithm, you can repeat the same phase retrieval calculation and you get the same image every time. You have to ensure you've set up the experiment right. You've got to understand your noise. There are things you need to know, but remember, when you make an SEM image, if your sample is at an angle, you also get an image that you don't really know about the sample. So and you can induce damage. And so, once again, there is no perfect imaging technique.
So the break
Yes.
was simply to acquire data on overlapping regions.
Great. That was what changed from where you could do a decent reflection mode microscope that you didn't need to have an isolated cell or nanoparticle, and it's even better than that because it overly the robustness, and then you add a few more things to tychography. You can do more tricks, like you can take a picture of your beam, which you can do, and that also then can constrain it. So there's so many simple additional constraints. And people talk about, what do you need to know about your sample? You don't need to know anything about the sample, but if you ensure overlapping illumination areas, and say you take a picture of the beam, then you can pretty much, if you can get scattered light, you can take a picture of anything.
In practice, how quickly do you have to do it?
Okay, so that's just how much photons you have to put in the big delay is that the commercial CCD cameras, which are very similar for X-rays as visible, because you have to take off the window and you have to back thin the silicon. In the X-ray region, in the visible, we have CMOS, so it's very fast. In the X-ray, nobody needed to transfer data fast. So you can take the image quick, but then it takes 30 minutes to read out the image. Yeah, so, but there are, the synchrotron sources are developing these very fast detectors because they're increasing the repetition rates for exfil light and synchrotrons. And so those detectors will become available. They're gorgeous, huge, big detectors for now. They cost $1 million, but eventually that'll come down to the commercial X-ray cameras, and so we'll be able to buy the same thing for $100,000, and then it becomes more feasible.
But you need your sample not to have changed in the time between taking the picture.
Or it's got to be a repeatable change, or else you do single shot, and all of that's possible. Yeah, that's right. You're absolutely right. And you need your sample not to change, and you need your illumination beam not to change, because you are assuming that is not changing.
Go ahead.
How long does it take for that single shot, and what are the factors that?
Oh, okay.
affect it?
So we are mostly interested in, nanosystems. Like this is a buried layer, buried interface imaging. So we've only recently implemented dynamic imaging. So we're, for all of this, it was sort of technique development showing we could do periodic objects. So we don't mind if the illumination is 30 seconds or 5 minutes or something, because we're totally limited by the data transfer rate from the camera. At the X-FELs, they do take single shot, but their jitter is very large. And at the synchrotrons, None of the synchrotrons are super fast right now, so they're doing static. For the most part, there's a few nice experiments that have been done that have looked at batteries charging and discharge, a nanoparticle battery. That was, I think, David Shapiro and Oleg Shpurko at Berkeley.
One question, what are the coherent requirements?
It is nicer if you have a coherent beam, because you can break down the scatter pattern and assume you have multiple modes. And actually, the synchrotrons have to do that because their beam shape can change. But that's a real pain because you've got to make it's a whole other set of unknowns. So by far the best thing is to have a mode that doesn't change. It doesn't have to be a great mode, but it should be, you do need coherence. And if you have it in a mode that doesn't change, that's by far the best to do. And in fact, that was my next, almost next view graph. But anyway, so you can see that it's getting more, we're getting more like a microscope from our little toy samples. The microscopes are very simple. It's just, you just focus the light gently onto the sample and then do this overlapping area scanning. And as somebody who kind of has worked in the X-ray region for a long time, you know, the good and the bad thing about refractive indices in the EUV is that they're super sensitive to any contamination. They change if you have a fraction of a monolayer, your EUV index will change, but that's great because you can turn that into very high contrast imaging. And in fact, it was this image that made us think that the technology was good. So this is an SEM image, just whatever SEM we had at JILA at the time. And this is the phase contrast image of the same sample. We weren't taking very good care of the samples, but this was nanostructures the students made on a silicon wafer. And what you can see is you can see all the same features on the SEM image as on the CDI image. But you can see that our contrast is much higher. And of course, the reason is one of them is a light-based phase contrast, and the other one is an amplitude contrast of electrons reflecting from the sample. And so, of course, if you're using shorter wavelength light, of course, you're going to get enhanced phase contrast. And so this was about 1.6 lambda spatial resolution in the transverse plane, and because we have phase contrast in the axial plane, it was about, I think this had about maybe 5 angstroms, okay, in the vertical. And we can, very long working distance. See, this is a very nice aspect of coherent imaging. You just have to, the spot size can be 100, well, maybe 10 microns, 1 to 10 microns. And so you don't need super tight focusing. And so it's really nice to have that capability. And in fact, this is a color map. This is the amplitude contrast, and this is the height contrast that we can, you know, get from that imaging modality. And then, of course, you have to check it versus AFM and just to make sure that one is really getting the same results and the same profile. And we do that very carefully. And then in this experiment, we took this is a damascene sample used by the semiconductor industry. So this is copper inlaid in an oxide and then coated it with 100 nanometers of gold. Our sputtering chamber is not that good, but in a visible microscope you see, excuse me, this was aluminum. So you only see, of course, the aluminum. If you use an AFM, it turned out that the sample wasn't completely flat, so the aluminum followed the profile of the copper. There was a little bit of, a fraction of a nanometer step height, so you could still see that the top layer was encoding the steps in the buried layer. But in these images, this is an amplitude image of the uncoated and coated sample. The amplitude image will only see the buried interface because it's the only thing giving you contrast. So the interesting thing here is if you look at these two images, they should look the same, but here the copper is bright and the oxide is dark. Here the copper is dark and the oxide is bright. So there had to be a change in the index. The only way that can happen is if there was material interdiffusion of aluminum into the copper. So it just showed that we could see the interdiffusion at that buried interface and get really high precision of, a nanometer or so. And this is as we were learning, how do we take the images. And then more recently then we wanted to show that we could image periodic samples. This is relevant for technological applications. And initially we got terrible results. So this was the SEM image, and this was our initial reconstruction. And it had all these features that weren't present in the scanning electron microscope image. And eventually we realized what was happening is that even tychography, the algorithm can get confused between features in the beam and features in the sample. And so we had to come up with another algorithm to stop. And so that's why you were getting these fringes. And so we came up with an algorithm that what you do is you take a picture of the probe with your CCD camera, just move the sample out of the way, take a picture of the probe, and then use that as the intensity of the probe, feed that into the algorithm, and suddenly you can stop it, getting confused between features in the illumination beam and the sample, and then we get a beautiful agreement. This is in reflection, and this is an SEM image in reflection. This is a coherent diffraction image in transmission. So, what's white here is dark here, and when you take that into account, we get a very nice high-resolution image. And in fact, this is the first sub-wavelength image at short wavelengths that anybody has been able to take using any light source. So it's really helping us push the spatial resolution limits. So this is our resolution with this. We call this a modulus enforced probe. And then because we have phase contrast, we can make a height map of this sample. And of course, we do all the checking of, is all our optics correct? Does the resolution make sense? We always have to do that when you're developing a new microscopy technique to make sure you're doing the right thing. And we were happy Dennis got the Division of Laser Science Thesis Award for this work. I mentioned before, it was one case where it was very fortunate that Henry had started taking the engineering of these light sources seriously. We couldn't make the images without a very stable wavefront. And so we struggled, and JILA has good machines, instrument builders, but we couldn't get the microscope to work using the university-built sources because, we're physicists, we understand the algorithms, but we don't understand the mechanical engineering. So it was a case where then when we borrowed an engineered system, then we were able to make the images because the pointing stability and the intensity stability of the light source was good enough that it wasn't another variable that in the imaging. And so interesting, this is the best image that anybody has made using his own plate. It's still the record. Using a spatial resolution of 50 nanometers using 13 nanometer illumination. So it was a Nature paper, and more recently an Optics Express paper. And so this is why, it's not just the tabletop light sources that are moving to this imaging modality. This is why all the synchrotrons are doing it and all the X-ray free electron lasers are doing it, because we can finally build a decent X-ray microscope. And, we're trying to generalize it, to be able to put in the sample at an angle. Not everything reflects light in the X-ray region, but everything reflects light at glancing incidence. So by making, because this is why the surface of a lake is a mirror at glancing incidence, and that's true also for X-rays. So we learned how to make the phase retrieval algorithms work even when you had this distorted diffraction pattern due to a conical diffraction, and you can correct for that. The wonderful thing, in a regular microscope, you have to build this gorgeous, spherical and chromatic aberration-corrected lens. With computational imaging, you can do all those corrections in the software, and so it's and we know how to do that now. And this is why you would want to do it. If you look, this is just an example at 13 nanometer wavelength, and this is all kinds of, metals, PMMA, oxides, silicon, and if you scan the angle, you can distinguish between all of these materials. So it gives you chemical and elemental mapping of, an arbitrary sample. Chrissy Porter is very interested in technological applications, so this is what she's working on now to show that we can really profile very complicated nanostructures and understand doping. Myself, I would love to, my interest is, can we see, spin currents or charge currents as they move across an interface. These are, very important for next generation electronics or, photovoltaics, where are the charges being trapped. So, but these samples are certainly easier than that, when we fabricate a nanostructure and we know what the doping is. It's a much easier sample for us to develop the imaging techniques on before we try to image a sample that might be changing in a way that we don't yet understand. Then it's hard to develop the technique and figure out what's going on in the sample. So what we've been doing then recently is taking all the spectroscopy that I talked about yesterday and seeing, can we make an image the material. The spectroscopy is wonderful. We can capture these attosecond lifetimes, but if your sample isn't uniform, that doesn't work. So it works great on a crystal, single crystal. So we can do it on charge density wave materials, on these magnetic materials, but can we do it on isolated nanostructures? And the first system we decided to look at was a thermal transport, thermal and acoustic transport, because that one has the time resolution we need is not super high. Picoseconds are fine. We don't have to tackle pushing everything to the limit at the same time. And so, and we know a lot about these materials. We've done a lot of spectroscopy trying to understand this nanoscale transport, or how the mechanical properties of materials change as we go to well below 100 nanometers. Coming to monolayers. How do the mechanical properties of material change as you add a monolayer at a time? And we would do a spectroscopy of this material. For example, we'd heat the nanostructure, the laser pulse comes in, and then, of course, if you heat a nanostructure, it's going to thermally expand, but if it's coming in now, and so you get it, yes. It thermally expands, but of course it's connected here on the substrate, and so the initial rapid thermal expansion will give rise to acoustic waves that travel into the material and then across the surface. And we can use that to characterize heat flow through nanoscale interfaces, or what the mechanical properties are of either thin films or the underlying material. And so, so we made a movie of heating one of these objects. So the idea is we have a laser pulse come in, heat a part of a nanostructure, and then probe, do tachographic imaging of that nanostructure. It's a fairly periodic thing, so we don't have a huge amount of diversity in the scattered data, and that's where this tricks on the algorithms, these modulated enforced probes, are very good, because it's more forgiving of lack of redundancy in the data. And then just do pump probe to see how that nanostructure is changing. Looks like a very simple experiment, a little V-shaped nanostructure. We're just going to hit it with a laser, as Misha was talking earlier. You just pop it with a laser and see what happens. However, the interesting thing is that you can't actually calculate this. It's actually computationally too challenging to, from a first principle, calculate the thermal and acoustic response of a V-shaped nanostructure. You can kind of do an approximation for a regular non-changing line, because you can imagine all the acoustic waves. If you have a uniform structure, you have some chance of being able to model the acoustic waves. In a non-uniform one, you're going to have a lot of dynamics happening. And so we could not do a full 3D simulation of the whole object, but we approximated this as a uniform line. And then we can do an approximate. So the students were laughing because it was quite challenging technically to make the nanoscale movie, but it turns out that at least they could make it. They couldn't do the whole computation. It's just we didn't have computers big enough. So this is the simple approximation. You can see the acoustic waves running in, the acoustic waves launched on the nanostructure itself. So after heating with the laser, you should see some waves both within the nanostructure and also, let me just run this one more time. You should see this depression here run out as you're launching surface acoustic waves in the substrate. And sure enough, we see that in the movie. Robert has a better movie, but this was an older version. But you can see all that same dynamics in the actual movie that do indeed correlate with these Rayleigh and other acoustic waves excited in the nanostructure. And of course, the interesting thing is this was all imaging that was done in the extreme ultraviolet region of the spectrum, in this photon energy region. As you go to soft X-rays, and we know how to make these, the penetration depth increases. So the ability to make a 3D functional imaging technique then as we move into the soft X-ray region becomes possible. And of course, it's all optics. Everything scales with the wavelength. So whatever, the current diffractive imaging techniques are now, I won't say mature, but they're sort of established. And so then it only becomes, do you have enough photons to illuminate in the time it takes? Are the photons coherent? Do you have enough of them? And you can scale. There are geometrical constraints that you need to fulfill. You have to oversample the diffraction pattern to get good agreement. But as detector pixel sizes go down, that also becomes easier. So I hope I've convinced you that we can push X-ray measurements in the time domain to the attosecond regime. X-ray measurements in full field imaging. This is not a fraction of the structure. It's the whole 100 micron by 100 micron type field of views. We can push that to below the wavelength limit. And the interesting thing is we compared our microscope to the best X-ray microscope that Zeiss can build, and we are about 10 to 100 times faster, even though we're slow. We're so faster than you can actually implement using a much more expensive traditional zone plate microscope. So it's kind of interesting. And so as part of the center, we want to add this to electron imaging. The interesting thing is that we can use the same laser to do nanoprobe imaging, X-ray imaging, and electron imaging. Working with Pietro Musumeci's group at UCLA, the same laser can generate bursts of electrons, light, and X-rays, visible light and X-rays. So it's possible. We are trying to build a hybrid microscope, but we're also working with the advanced light source and the advanced photon source to get some of the algorithms that John and Stan Osher at UCLA, he does a lot of algorithm development. So putting those on the facility, both the facility scale X-ray microscopes and the centers for electron microscopy also. So we're trying to build a legacy of new imaging techniques, new functional imaging techniques. So I think I've covered this in previous talks. So it would not have been the capability I would have predicted 10 years ago from this harmonic technology, because we had all been running down the high temporal resolution path. So it's a little bit surprising. I think we were just fortunate that these coherent imaging modalities are perfectly suited to the coherent X-rays, and that that it's a significant improvement compared to traditional zone plate microscopes. And so again, I need to thank the group for the work that came out of our group, and then Naomi, John, Roger Falcone, and these are all the students. This is everybody in our center. It was at our retreat, so it's about 100 people, but they come from industry, national lab, advisory groups, students of all kinds, outreach. It's a wonderful team of people, and a privilege to be able to work with them. And thank you so much for your attention.
Thank you, Margaret. There were already, of course, being quite a few questions, but we do have time for certainly a few more. Yeah?
All right, so two quick questions. One, are you guys relying much on machine learning in the construction algorithms? And secondly, for something that, let's say you're a starting investigator with a startup of, say,
Yes.
$500 million.
Yes.
How far can we get right now with what you're presenting?
Okay, so it depends. Let me answer the second one first, which is, for example, there's the cosmic beamline at the Advanced Light Source. It's based on, the cosmic stands for Coherent, and I forget what the other acronyms are. But, the national facilities are implementing these, and you have to write a proposal. There's a lot of work. Laura Waller and others are using this, and Jamyao, the same approach with visible light. So we're putting this in undergrad labs. So it's a great way to get computation into the physics curriculum, because you can do it with a laser pointer.
My question was like on the sources.
Oh, fine.
Like if
Okay.
I want the whole thing in my own lab.
Okay, but do you want an electron microscope or an X-ray microscope?
X-ray, yeah.
Okay, so if you want and it depends then what region. If you want 13 or soft X-ray, but let's say the whole thing, if I were to do a calculation, it might be 1 million. So it wouldn't be something that everybody would want in their own lab, but it's not crazy compared to an SEM or a TEM. It's another complementary tool. Yeah, but
I guess part of the question might also be not just the money, but also like if you started, how long would it take to actually get up and running and start to do some of the imaging you're talking about?
So so I think it'll be ready for prime time in maybe the simplest version, not in the X-ray region, in the extreme ultraviolet. That would be packaged for. I mean, this is just published last year and this year, so nobody has engineered the microscope. So our students literally take a little bit of extrusion and put the sample on it. They have to put it on a really good stage because you've got to scan with nanometer precision. But it's no different from taking any imaging technique and adding engineering to it. So the engineering has been done on the source already, but not on the microscope.
So right now it's a sample scanning modality.
Yes. Both for reflection and for reflection, you always want to scan. Right? And then the machine learning. So not enough. We need to do more, because so far we've been doing technique development. So speeding it up has not been a priority. However, there's a very nice company, Phase Focus in UK, that came out of Rodenberg's group, who developed tychography. They're doing it for cell sorting and bioimaging, really amazing phase contrast, and not just at the edges. So you get the very nice phase contrast everywhere. And they have packaged software systems that you can buy. We tend not to do that because we want the students to understand the details, because we have to, because if we're doing these glancing incidents and such, we have to be able to modify the algorithms. And so we do our own, and they're clunky and slow, but that's what we do at university. We're not trying to optimize the speed so much, but clearly for the tabletop to have impact, they'll have to be engineered, and they'll have to be packaged so that people can use it like an SEM. Yeah, and that's not an issue. Somebody has to do it.
Any further?
Could you talk about what are the physical limits for X-ray
Ah,
imaging?
so John and I talk about that a lot. So it's not crazy to think about atomic resolution X-ray. It's possible, maybe not on every sample. It's not something we put down in our strategic plan, but it's not. It's something. It's something that we can still try and do. So we'll. But of course, it probably isn't the thing that will. We have that with the electron. So it's the chemical, elemental, time resolution with very good spatial resolution is probably the thing that will have impact on many materials. All right. And to be able to look at reactions at buried interfaces in energy systems. In thermoelectrics, for example, there's a lot of work on nano-enhanced thermoelectrics, but very, very poor ways to probe them right now.
And as a follow-up, you mentioned packaging this so it can be more easily used. What do you see as sort of the limit on the size of the system?
Oh, like for example, an SEM type size, yeah. It depends on the application. It depends how fast you want to be able to image, but that's sort of the size that would be practical. You don't want to try. It doesn't make sense to try to compete with an XFEL or a synchrotron. We have those, right? So you want to. There'll always be this trade-off of you can do it in your own lab every day. It might take 5 minutes, but who cares? Because it takes a lot longer to get on a plane and fly to the synchrotron. So.
I was wondering, Margaret, this is obviously wonderful, technology and just really great. I'm wondering, is there a problem you're sort of dying to use this, know the answer to and really use this technology for on a, sort of five-year time scale or something?
Well, in these magnetic materials, nobody knows the nature of the light excitation, and with an ability to do dynamic image profiling, we can distinguish between all the different mechanisms that might be giving rise to these very fast responses. We can see if there's spin currents that are piling up at interfaces or where. So that would be one example of a sort of an interesting physics question that might be hard to answer using other techniques, because you have to be able to probe an opaque material, and ideally you would like to have multiple interfaces that you could look at as to what is happening at each of those. And then we're also looking at all the usual things. People are looking at skyrmions and magnons and all that. Yeah.
So from everything what you said, so I understand that one of the next big frontiers in that direction is actually the coherent manipulation. And so I guess that's probably a little bit more than a 5-year time scale. But so what would be the questions there? So PIT or quantum information or deterministic chemistry to all levels. So
So even something simple that we're hoping to answer soon. This is maybe it's a different answer to Lena's question, which is people don't even know how phase changes happen. They don't know if it's a phase separation, or that often there's they're not abrupt. Theoretically they're abrupt. Experimentally they're not. So whether does it start at a defect? How does it start? Is it homogeneous? Both laterally and axially.
Involve the timing, not just
no, the imaging and the timing. Imaging and timing, because a lot of these materials are not there was a beautiful Nature Materials issue where her name is, she's a material scientist from Stanford, and it was this lovely Nature Materials issue on imaging. And, she sort of put it very well that we have to get away from imaging one tiny piece of a sample with a different technique and then publishing a paper on it, because you actually don't know if you actually understood the material at all. They're very complicated. And so what she was saying is we got to do some much more correlative imaging and such. So I think even though, you're right, could we image every aspect of one of these strongly correlated materials? I think, yeah, that's going to happen. I don't know how long, but it's going to happen. It may not be our group, or it might be. There's lots of people looking at it, but I think there's even more lower hanging fruit that would actually have a big impact on material synthesis. I mean, so this is the issue, you know, that there's very few characterization tools on, you know, growth chambers. So this is what Ian Fisher keeps saying, you know, that you make a tiny change in your growth of many of these advanced materials and the properties will change. But we don't have good ways of being able to look at that happening. So there's a whole pile of very low-hanging fruit that would be good, as well as all the beautiful physics, which I love.
Well, thank you so much.
Thank you, Lena.
For this
Thank you,
wonderful
everybody.
lectures.